today's edition about GIDEON, the Global Infectious Disease and Epidemiology Network, I want to explore how to identify and diagnose pathogens and microbes on their characteristic behavior on different media, enzymatic tests, looks, morphology, and so on. GIDEON is a very powerful tool, much better than the original dichotomous tree. And we have a certain set of plates, tests, what we usually do, and that gives you a pretty good idea of what your organism, your unknown, is. And this can be done with the unknown project in the lab, and it can also be done with the adopter microbe if you're taking the labs online. These are the media and tests, enzymatic tests and characteristics, what we look for when we try to diagnose and identify our organism. You can see it starts off with the Gram reaction with the cell shape. We look whether it grows different oxygen requirements, if it needs oxygen to grow, or if oxygen is toxic to it. We look at the blood agar plate growth. We can also look for hemolysis, look at McConkey agars. We look at growth and lactose fermentation. We look at two enzymatic tests, oxidase test, catalase test. We look for glucose fermentation in the phenol red broth. We look for motility, enol production, and sulfite production. And then we also have DNase plate, where we look for the enzyme DNase produced. We look for some pigmentation. We have the mannitol salt agar plate, where we can see whether the organism can grow on salt and also ferment mannitol. So these are our key tests, what we usually do. But there are obviously many other tests. This is a small selection, and I'll show you how once you know the results, how to find out the results for your organism. Again, in the lab, if you're taking the lab, you would actually do these experiments with an unknown organism. If you're in the online environment, you have your adopter microbe, and you can find out what the characteristics are. But let's go over to GIDEON and let's explore it a little bit. Again, today we mainly talk about the lab part, where we go to the diagnosis of bacteria. We're not looking at mycobacteria or yeast, we just focus on bacteria. And I prefer the classic version of the diagnosis because in the interactive version, although it is very interesting and it's more like the old-fashioned phylogenetic dichotomous tree, it has a lot of tests what we don't do, so you always have to skip certain steps. So again, let's go to the classic version and then look what we have. Let's have an example. So for example, we have an organism, and that's one of the activities we usually do. We say, who am I? And we give you the characteristics and results of these tests for a specific microbe, for a specific bacterium or pathogen. And then you have to find out what the organism is, how it compares to some of the closely related organisms, what other tests can be done in order to make sure that you know exactly what organism it is. Because that is really important for treating patients with certain antibiotics as well. So it's really important to find out. Okay, let's get started. Here's an example. We know that the organism is Gram positive. It is a bacterium in pairs or chains. And then you also can see how it eliminates answers if there's no organism that fits these criteria, which is very nice. It's not an acid-fast bacterium because it is Gram positive. And it grows as a facultative organism. That means it grows with and without oxygen. So in the anaerobe chamber, it grows fine on the bench, and it also grows in the anaerobe chamber. It definitely grows on blood agar, which is very common. It does not grow on McConkey agar because McConkey agar is mainly for intestinal bacteria, but it contains biosalts. And so organisms that are not exposed in their normal environment to biosalts usually don't grow on it. Some do. It is oxidase negative and catalase positive. These are two enzymes, and again, you can always hover over the test and then see what you actually do for for the tests. So it's quite nice to have all that information there. Oh, let's make it catalase negative. So oxidase negative, catalase negative. It does ferment glucose. And then we go to some of the next tests here, the general tests. It does do beta hemolysis, and which is seen on the blood agar plate. It does not produce coagulase. It is not motile, and it produces indole. And I think these are the main ones. No hydrogen sulfide. DNA is positive, and there's a few more, but these are the key ones. Salt, it does not grow in salt environment. And then here you get fermentation acidification of different carbon sources as well. So you have a lot of different choices. And we had the lactose, but it didn't grow in McConkey agar, so we don't know. And it did not grow in salt either, so we don't know about the mannitol. Alright, so let's see what we have. So our top results are Streptococcus pyogenes, Streptococcus pneumoniae, and angiosis, and a couple of other ones. So there all seems to be definitely in the Streptococcus area in the genus, but we don't know exactly what it is. And if you can look at the probability, it seems very likely for Streptococcus pyogenes and very little for the other ones. Let's see how they're different, because it's really hard to sort of know what's the difference between Streptococcus pyogenes and Streptococcus pneumoniae. Obviously they live in different environments, but let's see. So GIDEON has this great function where you can compare the two results, and then, or you can do more than one, and then it gives you the results here. So we had Streptococcus pneumoniae and Streptococcus pyogenes, and now let's see. So this is always the same here, that's why they're so similar. And then here beta hemolysis, so pyogenes, and that's probably why I put pyogenes on top, is because we said it was beta hemolytic, and it doesn't do coagulase, and ONPG beta galactosidase, that's often as well. So that's another test we could do, but we haven't done this test. So I think right now the hemolysis is our best indicator that it's Streptococcus pyogenes. What you have to consider with bacteria is that not every isolate, not every strain behaves the same way. So the different indicators here, for example, always, that's in all isolates that have been reported. Usually it's in most, 90 to 99% of the isolates, and then you go down to 1 to 9% and rarely, and then never, which has never been reported. So again, not each organism or not each species is the same as it performs on these different tests. Okay, so now we understand what the difference is between these two organisms, and we can go back to our test. So pretty sure that it's Streptococcus pyogenes. Let's figure out a little bit more about it. Let's click on it, and what is it? It is common in, it's a pathogen. It's in pharyngitis, found in the mouth and oral cavity, but it can also be transmitted through oral lesions during dental procedures. It can also cause rheumatic fever if it gets into the body in the bloodstream, and a bunch of different other infections. So sometimes it's really hard to associate, especially oral bacteria, with other diseases throughout the body. You can look at the drug susceptibility. There are some synonyms, and then you also have some images, what it looks like on a plate growing, on blood agar in this case, and then here you have your full phenotype. So again, if you work on the adopter microbe, you want to go to the phenotype of your organism and then find the results to add it to your table and the results as well. So again, that's how you identify the organism. Again, you'll see this in different forms of the lab. There's definitely activities of who am I, where we give you either images or we give you a description, and then you have to sort of figure out what you're working with. All right, again, we're working with microbes, bacteria only, and the way you get there is to go to lab, bacteria, and then classic identification. It's pretty fun, it's really interesting, and there's a lot of interesting facts what you can find out about the organisms, and also distinguish between these two related organisms or two organisms with a similar profile. Okay, that's a big part of here, adopt a microbe.